Okay, so welcome to this next video in the playlist on the cardiovascular system. In this video, we're going to talk about the regulation of eNOS by two proteins known as NOSIP and NOSTRIN. Okay, so firstly what I'm going to do is I'm going to talk a little bit about what eNOS is, just to give it a little bit of a context to this, and then we'll talk about what NOSIP and NOSTRIN are and how they both downregulate the function of eNOS. Okay, right, so let's draw our picture of the layers of the blood vessel. So, if this is a blood vessel here, okay, right, then the innermost layer of the blood vessel, which lines the actual lumen of the blood vessel, is a layer of endothelial cells. So I'll draw a layer of endothelial cells here. So here's one endothelial cell, 2, 3, 4, 5, 6, 7. Okay, right, so these are these endothelial cells which are facing in on the actual blood contents of the blood vessel here. So I'll draw their nuclei here. Okay, so these are these endothelial cells. Now, these endothelial cells are sitting on a layer of collagen known as the basement membrane. So I'll draw this layer of collagen here in turquoise. Okay, and the basement membrane, this layer of collagen, along with the layer of endothelial cells, together, this is known as Tunica Intima. Okay, so this is called Tunica Intima. Tunica means layer, intima means close, so it's the layer which is close to the actual blood within the blood vessel. Okay, so around the Tunica Intima you then have the Tunica Media. So the next layer is the Tunica Media, which is a layer of smooth muscle cells and also elastic tissue. So the smooth muscle cells are basically arranged in circles. What you'll have is a circle of smooth muscle cells, like so. So one smooth muscle cell, then the next smooth muscle cell, the next smooth muscle cell, here's the next one along, then the next one, okay, and then finally completing the circle like this. So you've got these circles of smooth muscle cells, and I'll just colour in these smooth muscle cells in a pink colour so that we can see them, okay. So, when these smooth muscle cells contract, what it's going to do is they're going to become, their length is going to decrease. And when their length decreases, then that's going to mean that the circumference of this entire circle of smooth muscle cells is going to decrease. Now, if the circumference of that ring of smooth muscle decreases, then the diameter of the ring of smooth muscle also decreases, which means that the diameter of this the lumen of this blood vessel is going to decrease. So, what's basically going to happen is you're going to get vasoconstriction. The diameter of the lumen is going to constrict, and there's going to be less room for blood to flow through. So, these smooth muscle cells lining the blood vessel, these control how thick or how wide the lumen of the blood vessel is. Okay, and this layer of smooth muscle cells along with elastic tissue, which is also circular in origin, circular in orientation, is known as Tunica Media. Tunica again means layer, and then media means the middle layer. Okay, and then finally the outer layer of the blood vessel is a layer of connective tissue. So I'll highlight this in yellow. Okay, so in yellow here we have this outer layer of connective tissue, and this basically, well, it forms another layer around the blood vessel that holds everything together, but its role is also to connect the blood vessel to other structures in the body and hold it in its native position in the body, basically. Okay, so this final outer layer of connective tissue, and let me just tidy it up a bit down here. Okay, so this final outer layer of connective tissue is what's known as tunica adventitia. So this layer here of connective tissue is known as tunica adventitia. Okay, right. So, these endothelial cells here, They were once believed to just be the lining of a tube. They were the inert lining of a tube. That's what we used to believe they were. Their only job was to control what could move out of the blood vessel and what stayed in, basically. They were just controlling permeability. Now what we know is they have a much more active role than that. They are a gland, effectively. They are secreting all sorts of hormones. And most importantly, arguably, they secrete nitric oxide. Okay, so they secrete nitric oxide here. NO. Strictly speaking, nitric oxide should be called nitric monoxide, but everyone calls it nitric oxide. So they secrete nitric oxide, which diffuses out to the smooth muscle cells, which are more peripheral to the endothelial cells, and causes those smooth muscle cells to relax. So the endothelial cells are continuously controlling how wide the blood vessel is, basically. So they play a very active role in the cardiovascular system. Okay, so the enzyme which produces the nitric oxide is within the endothelial cells, and this enzyme is known as endothelial nitric oxide synthase. Nitric oxide synthase, or often abbreviated just to eNOS, basically. So you take all the initials. So this is often abbreviated to eNOS. Now, more correctly, they should be called nitric oxide synthase 3. The reason it should more correctly be called nitric oxide synthase 3 is because it's found in many other places than just the endothelial cells. So more correctly, this is NOS3, nitric oxide synthase type 3. Okay, but everyone usually refers to it as eNOS. Now, this enzyme is it's anchored basically to the membrane of the endothelial cell. So what you find is that if we draw an endothelial cell here, it's basically positioned in the membrane of the endothelial cell. So let's show it here. So here is our eNOS enzyme, drawn as a box here, and it's anchored to the membrane of the endothelial cell. Okay, and the way that you achieve it, achieve this feat, that it is anchored in the membrane, is that you add palmitoyl and myristoyl groups onto it. Okay, so let me just discuss with you a bit more in depth what the structure of eNOS is and where these palmitoyl and myristoyl groups are added onto. So, eNOS enzymes actually function as a homodimer, so they don't go around on their own. So I'll show you the structure of one eNOS protein firstly. So here's an eNOS protein. Okay, well, at least this is my cartoon of what an eNOS protein looks like. So here's the carboxyl terminus of the polypeptide, and here's the amino terminus of the polypeptide. Now, this circle that I've drawn here is supposed to represent the oxygenase domain of the enzyme. So this is the oxygenase domain. Okay, and this down here is the reductase domain. Now, in fact, to make nitric oxide, all you need is a single oxygenase domain and a reductase domain. So you might ask, well, why can this enzyme not function as a single protein then? Why does it need to sit in homodimers? And the reason is that the oxygenase domain and the reductase domain are not in the correct positions relative to one another to actually function. So what you do is you take another eNOS protein here, okay, and this one will have its oxygenase domain coming back here, and you dimerize them together. So this is the dimer of the eNOS proteins now. Okay, so here's an oxygenase domain, and here's a reductase domain. So I'll colour in oxygenase domains blue. Okay, so this is an oxygenase domain. This is also an oxygenase domain here. Okay, and then in red I'll have the reductase domains. So this is a reductase domain here, and this is a reductase domain here. Okay, now what's going to happen is this oxygenase domain of, let's say, eNOS protein 2 is going to function with the reductase domain of enos protein 1. So these two together are going to make a functional enzyme. Okay, so this is going to be a functional enzyme. And then these two together, the oxygenase domain of enos 1 and the reductase domain of enos 2 over here, they are also going to make a functional enzyme together. So you dimerize them together so that the oxygenase domain can have the correct position relative to the reductase domain, and they can function together, basically. Okay, that's why they need to form this homodimer like so. Right, so where do we add these myristoyl and palmitoyl groups onto, and what does that even mean? Well, we add them on upstream, basically, of the oxygenase domain. So the polypeptide, prior to you getting onto the oxygenase domain, we add palmitoyl and myristoyl groups. So what does that mean? Well, let me firstly talk to you about what myristic and palmitic acid are. So myristic acid. Myristic acid is the old name for a carboxylic acid which has 14 carbons. So here's the carboxylic acid group. That contributes 1 carbon. We then need 12 methylene groups. Now, I do not have the space or the time or the inclination to draw 12 methylene groups out. So there is a trick. You can put brackets around this like so. Okay? And you can say, copy that out 12 times, okay? And then finally, you draw the final one, because that's not a methylene group, so it's a methyl group. So overall, that has 14 carbons. You have 12 methylene groups, so 12 carbons there, and these two here to make 14. That is the structure of myristic acid. Okay, palmitic acid is very similar. Palmitic acid, okay. Palmitic acid means the 16 carbon carboxylic acid, again totally saturated, which means that it has as many hydrogens off these carbons as it possibly could, i.e. there's no way you could build a 16 carbon carboxylic acid that had more hydrogens than this one. Okay, so this is the methylene group. This time we're going to have 14 of them, and then on the end we'll have our methyl group over here. Not a single double bond in sight between the carbons, at least, anyway. So this one has a 16 carbon chain backbone. 1, 2, and then 14 here, 16. So, when we add myristic acid and palmitic acid groups onto the protein, they are then said to be myristoyl groups. So, this is some fancy terminology. Myristoyl groups and palmitoyl groups. Okay, palmitoyl groups. And the process of adding myristoyl or palmitoyl groups onto a chemical species is known as myristoylation, which is a fantastic word. Myristoylation. And then similarly for palmitoyl groups, it's palmitylation. Palmitylation. Okay, right, so big words for processes that are very simple. Right, so what are we actually going to add these onto? Well, we're going to add them onto R groups of amino acids in the polypeptide upstream of the oxygenase domain here. So you add these on, and basically what ends up happening is you get long chain carboxylic acids, which are very hydrophobic, being added on to this protein. So here are these long chain carboxylic acids that have been added on to this protein here. Okay, and basically these are going to stick out from the enos homodimer, and they're going to allow you to implant the enos homodimer or suspend the enos homodimer in the plasma membrane, because these hydrophobic tails will want to stick into the membrane. They'll want to interact with the long chain carboxylic acid tails of the phospholipids that are in the phospholipid bilayer. Okay, so this is the way that enos can suspend itself at the plasma membrane. There are other ways that it can suspend itself at the plasma membrane. So specifically, these palmitoyl and myristoyl groups are very good at isolating enos into caveolae. So here's a caveolae, one of these little indentations of the membrane inwards. Okay, so this is a caveolae. So all cells are covered in these. They have little invaginations of their membrane. And in these caveolae membranes, what you have is the protein caveolin 1. In endothelial cells, at least, you have the protein caveolin 1. So this is caveolin 1. Now, eNOS can also bind to caveolin 1. Okay, so here is caveolin 1. So that's another way you can suspend it at the membrane. And finally, also you can bind it to certain GPCRs. So for instance, the bradykinin 2 receptor, the B2 receptor, this can bind eNOS to its carboxyl terminal over here. So eNOS can bind to the carboxyl terminal of bradykinin 2 receptors. So there are many different ways to suspend eNOS at the plasma membrane, and this is very, very important for eNOS function. It functions best, it responds to calcium signals best if it's at the plasma membrane. The function of NOSIP and NOSTRIN is to downregulate the function of eNOS and make the endothelium produce less nitric oxide. Okay? And they work by stopping it from suspending itself at the plasma membrane. And we'll discuss this further in the next video.